already open, do it. Ali and I have known each other for more than 2 decades easily. Yeah, gray beards, both of us. I'll give you a little bit of background. I'm not going to give you all of his background because that will just take way too long. He has so many accolades. But I will tell you that in those 2 decades, I can candidly tell you that as a trainee in his lab and all the other trainees, you are in a good position. But I also know that Ali selects very well. So I already know that all of his trainees, including you, Hal, are excellent. Ali did his MD at Queen's University, then did a bunch of internships, including an interesting rural internship residency, did some volunteer work. Then he did, I guess residency isn't the correct word for some of those things, but so I apologize. But then he did a proper residency in hematopathology. He is now Distinguished Scientist, Michael Smith Genome Science Center at BC Cancer Research Institute, which I suppose is his primary affiliation. He is professor in the Department of Pathology and Laboratory Medicine. He is Tier 1 Canada Research Chair in blood cancers. He has many other affiliations, including, and most importantly, being a member of the Center for Blood Research. And I thank you for joining us.
Thank you, Ed. Much appreciated. This is on, right? You guys can hear me? So I'm not really going to speak very much. I just want to give a little bit of a background of what my lab's interests are, a little bit of background on the diseases we study, which are MDS and AML, and then hand it over to Hale Kincross, who is a year 5 PhD student in the lab. He's done an outstanding job, and he'll tell you about some of the work he's been doing. So we're interested in the myelodysplastic syndromes and acute myeloid leukemia. These are both very aggressive myeloid cancers. They arise in the bone marrow, and they present generally with low blood counts, which means you might have infections or you might feel fatigued because your hemoglobin's low. And with respect to the myelodysplastic syndromes or MDS, what's happening is that within the bone marrow, you still have a lot of cells. They're just funny looking. They're not doing what they're supposed to do. And so what happens is in the peripheral blood, you don't get your cellular output that you're supposed to. And the progression of these diseases in general is either to bone marrow failure. In other words, you're just not producing blood that's functional, or you get an even more aggressive cancer called acute myeloid leukemia. And that leukemia is the most common acute leukemia in adults. It's, as I say, more aggressive than MDS, also presents in the same way. And despite intensive chemotherapy, bone marrow transplant, resistance to therapy is common. Patients are refractory or relapse after going into remission. And so a major focus of the current focus of the lab is trying to understand why patients don't respond to therapy. And part of that, to understand that, is to understand how the disease works in the first place, which is what Hale will be talking about. So here, just as a comparison between lung cancer and MDS, and what you see is that adjusted for stage and age, outcomes in MDS are worse than in lung cancer. And everybody knows that lung cancer is a bad cancer. And similarly, in acute myeloid leukemia, overall survival, a 5-year survival, is less than 30%. It's better if you're younger than if you're older, but in general, if you stratify, you can stratify patients based on their genetics, so mutations or translocations that they might have. And so if you have favorable genetics, you might do a little better. If you have poor genetics, you're going to do worse. It's also these survival probabilities are also dependent on age, as I mentioned. When you look at the number of mutations that are present in AML, compared to all other cancers here, what you see is the AML actually has very few mutations compared to many other cancers. They're in the area of what childhood cancers have, which are low. But nevertheless, the types of mutations that do occur are recurrent and can be organized into different classes, as you see here. So there's mutations in genes that affect DNA methylation. There are tumor suppressors. There are those that down here that affect splicing, and so on. A number of years ago, what graduate student in the lab, Rod Docking, did was we sequenced a whole bunch of AML patients, close to 200. And he used this, we were trying to create a clinical assay that could be used for patients. And what Rod did was to develop an expression-based signature. So this is not based on mutations in the genome, but rather what RNA species are being expressed. And he developed a score that, as you can see, we called it the AML prognostic score. And you can see this is a volcano plot. And you can see in our own training set, obviously he did very well. But when we looked at two other independent data sets, the APS score was as predictive as age or P53 mutations, which are very predictive for a poor outcome. And they do better than a leukemia stem cell score. So this is in two independent data sets. And so the question was, if we're going to use this clinically, we can look at gene expression and have a score, but current stratification stratification tools use genetics, so mutations and translocations. And so the question was, can you actually use RNA? So this was a number of years ago, and more people are doing this, but at the time, nobody used RNA sequencing to call genetic mutations. But what we found was that compared to this RNA-seq, compared to whole genome sequencing or whole exome sequencing, you can call variants at the same variant allele fraction as DNA-based testing. And in many cases, because the sequencing is much deeper, you actually call mutations that are missed when you sequence the whole exome or the whole genome. And so with this knowledge in hand, we looked at the mutations that we found that were recurrent in these 200 patients to see if there were mutations that had not been described. And what we saw was that there were a lot of genes in the ubiquitin system, and Hale will tell you more about what that is, that were present, but they weren't the same genes. So if you look at all the genes, there's a whole lot of genes that are mutated, but sometimes only once or twice. And so this has never been put down as a classification in AML. So really what we're saying is that mutations in the ubiquitin system genes are probably their own class of mutations. The only one that's previously been really characterized is Sybil, which is often found in different kinds of myeloid cancers. And so we focused on a gene called FBOX11, which was recurrent. We saw, I think, 4 or 5 cases of it. But what was more important is if you look at gene expression and you look at FBOX11 expression in AMLs versus normal hematopoietic stem progenitor cells, what you see is that FBOX11 is the most significantly underexpressed gene in AML, within this specific type of ubiquitin system. And the mutations that we saw that I showed you in the previous slide were all loss of function mutations, suggesting that loss of FBOX11 function is playing a role in myeloid cancers. So we asked that question. We noted that FBOX11 mutations were associated with this translocation, this AML ETO translocation or translocation of chromosome 8 with chromosome 21, and also with KRAS mutations, activating mutations in KRAS. There was published data that's saying that if you put both of these things into normal blood progenitor cells or stem cells, you couldn't really produce a leukemia. And so we asked, since these things were associated, if we actually knock down FBOX11, shown as F here, and we combine that with these two genetic events, could that cause AML? And so what you see is when we transplanted cells that were knocked down for FBOX11, but also had this translocation and this activating mutation, those animals started to die. Whereas if you just had these two things, or if you had a control empty vector, those animals survived. And what you got was a myeloid cancer that infiltrated the brain, the CNS, which is shown here. And that's just a myeloperoxidase stain showing that they are myeloid cells infiltrating the meninges of the brain. And so with that, I'm going to hand it over to Hale, who will tell you about all the wonderful work he's been doing.
Okay, thanks, Ali, and thanks to everyone in the CBR for having us out today. So with Ali now teeing that up, you see we have this connection now of the ubiquitin system being dysfunctional, likely in AML, and contributing to the transformation of normal blood and stems, or normal blood stem and progenitor cells into a myeloid malignancy. So we, as Ali said, we focused on this one gene here called FBOX11, which encodes a substrate recognizing component of this E3 ubiquitin ligase complex. So there's 40 different FBOX proteins within the genome that target will assemble into this complex and then target all different kinds of proteins for ubiquitination, which will lead to protein degradation or changes in that protein's activity. So we validated in normal blood stem cells, as well as primary AML bone marrow, that FBOX11 protein was depleted. And then we also validated that in CD34 cells isolated from adult bone marrow. So both cord blood derived and adult, that relative to AML, these normal healthy cells have much more FBOX11 protein. So we performed some sequencing to look at the whole transcriptome of normal HSPCs. So we had depleted FBOX11 and tried to identify the transcriptional programs that were likely changing in these cells. So we could see that among the top enriched pathways were these HSC and LSC gene sets, as well as these quiescent phenotypes came up as well in the upregulated genes. Contrary to this, in the downregulated were these slightly more mature progenitor gene sets, as well as ones relating to erythroid differentiation, which had been described for FBOX11 function before. And then also a bunch of mitochondrial pathways, which was something we hadn't seen before in the literature. So we took our FBOX11 depleted HSPCs derived from cord blood. We put them in liquid culture, and we could see that just a few days in culture here that these HSPCs that have been depleted for FBOX11 were retaining their CD34 positivity, as well as failing to proliferate, so they were remaining in a low cell number. And this we attributed to them being stuck in this G0 phase of the cell cycle, so they were unable to proliferate, which is supported by the transcriptional data suggesting these primitive programs. Immunophenotypically, using markers that have been described by John Dick's lab at the University of Toronto, the CD34 positive cells were displaying more of this HSC-like immunophenotype as opposed to the classical culture progenitor immunophenotype as well. If we look a little later in the culture, seeing what kind of output these HSPCs have, we could see that the FBOX11 depleted ones were depleted for differentiation into megakaryocytic and erythroid lineage cells, and this was at the expense of myeloid bias, which is a classical feature for like a myeloid cancer. When we plated these cells for colony-forming cell assays, you could see that relative to control, we completely lost these erythroid lineage colonies, with retention of only the colonies that remained were basically essentially all myeloid, but also lower in number in the primary CFC assays. When we replated those, we could see that there was an increased number in the secondary replating, so that suggests an increased stemness of those cells that did remain. So with FBOX11 being a E3 ubiquitinase, we performed an assay where we tried to identify what is the target of potentially that FBOX11 is not regulating when it's lost in AML. So we performed this mass spec assay where we tried to identify co-amino precipitating proteins with a flag FBOX11 in K562 cells. And we performed this with and without endonuclease treatment to disrupt potential RNA binding interactions. And we found that between these two assays, we found the 3 members of the SCF complex, so FBOX11 itself, as well as SCIP1 and COL1, and then 2 targets that hadn't been described before for FBOX11. So we just validated, we couldn't validate this that5 interaction, but we investigated LOMP1. So we performed a co-IP in our FBOX11 knockout line, or a knockout line with FBOX11 reconstituted. And we could see that when we performed a LOMP1 IP here, we could also co-immunoprecipitate FBOX11. And inversely, if we immunoprecipitated FBOX11, we could pull down LOMP1, validating that the interaction between the 2 proteins that hadn't been described before was likely real. So LOMP1 is a mitochondrial chaperone and protease. And remember, I told you we saw disruption in these mitochondrial transcriptional programs in our healthy HSPCs that had been depleted for FBOX11. So we tried to figure out what could this potential interaction with LOMP1 be leading to. So the first thing we checked was whether depletion or knockout of FBOX11 in K562 cells led to an increase of LOMP1 protein. And we found that it didn't change LOMP1 protein expression levels at the whole cell level. So what this suggested to us is that this LOMP1 or some interaction with FBOX11 and LOMP1 isn't leading to LOMP1 degradation, which would classically be through the proteasome. So that gave us a question of where and what is this interaction actually doing between these 2 proteins. So to try to figure out where these 2 proteins were interacting in the cell, we performed a proximity ligation assay, which you incubate your fixed cells with antibodies to your target. So in our case, FBOX11 and LOMP1. We then incubated with these oligo-tagged probes as secondary antibodies, performed a ligation of those probes, and then an amplification step where you get this rolling cycle amplification with fluorescently tagged nucleotides, producing a fluorescent spot you can detect on microscopy. So in our endogenous wild-type K562 cell line that has FBOX11, we see FBOX11 and LOMP1 indeed are interacting. When we knocked out FBOX11, those interactions disappeared. But if we put our flag FBOX11 back into those knockout cells, the interaction came back. And we quantified roughly where those interactions were taking place in the cell relative to the mitochondria. And we could see that they were essentially all within the cytosol, so not occurring in the nucleus and not occurring in the mitochondria, although some did occur very close to the mitochondrial surface. When we look at the topology of these 2 proteins now, so we could see that in, again, our fake K562 cells, FBOX11 was primarily in the, almost entirely in the cytosol with no mitochondrial localization. LOMP1, which is nuclear encoded again, you could see throughout the cytosol at low levels, but highly concentrated within the mitochondria here, overlaid with TOM20 in yellow. And then we saw a number of puncta of FBOX11 and LOMP1 sort of co-localizing really concentrating near the mitochondrial surface. So we knocked out, or we still had our FBOX11 knockout cells, and we looked at LOMP1 localization in these cells, and we could see that it didn't look quite right to our eye compared to the wild type FBOX11 cells, where we see a mostly co-localized with the mitochondria. LOMP1 in red was sort of getting trapped outside the mitochondria. But if we put our FBOX11, our flag FBOX11 back into these cells, suddenly it looks much more, we see much more co-localization of LOMP1 to the mitochondria. So to confirm this, we performed some, sorry, I have my quantification there. To confirm this, we performed differential centrifugation to extract the mitochondria from these cells. And we could see that relative to our knockout cells expressing empty vector, the reconstitution of FBOX11 almost doubled the amount of mitochondrial LOMP1 in these cells. Inversely, we took a different cell line and depleted FBOX11 by shRNA, and we could see that we had about a 65% decrease in mitochondrial LOMP1 when we did this. So that was telling us that this E3 ligase complex allows a modification of some kind for FBOX11 to promote LOMP1 into the mitochondria where it would normally be active and functional. So we were asked along our journey to get this research published by a reviewer to validate some LOMP1 mutants, which wouldn't be able to bind and interact with FBOX11. And that's pretty hard to do when it's a novel interaction. We didn't have confirmed where exactly those two proteins would be interacting within their independent domains. So we had a bioinformatician in the lab named Pranav who was able to model this using AlphaFold 3. And you can see LOMP1 predicted in red, as well as the SCF complex, so FBOX11 in this dark blue. And Pranav plotted the change in the surface area, the solvent accessible surface area of the proteins, to determine what inner specific amino acids of LOMP1 were becoming excluded or disrupted, to indicating a binding with FBOX11. So we could see a high concentration of this interaction between FBOX11 and LOMP1 right here at the N-terminus. We saw a couple lower confidence predicted interacting regions throughout the middle of the LOMP1 protein, as well as this very strong and two interacting peaks here within its LOMP1's ATPase domain. So based on this modeling in silico, we cloned two LOMP1 mutants to delete these two regions of the LOMP1 protein independently, and then express them in our K562 cells. So you could see, and we had attached a flag tag to the end of the protein so we could differentiate it from the endogenous LOMP1. So you could see that when we express these constructs, we are able to get expression that we can detect on a Western blot for these two mutants, as well as our wild type LOMP1. And we performed a co-IP. And as we had seen previously, when we IP'd this flag LOMP1 now, we could co-IP FBOX11, so that the two proteins were able to interact under non-denaturing conditions. But if we, for either of the mutants, we weren't able to see any FBOX11 interaction, suggesting that by deleting these domains, we were disrupting the interaction between the two proteins. As I had just shown you, we then tried to figure out, is this LOMP1 protein then mislocalized when it has either of these mutations? So relative to our empty vector here, we could see FBOX11, or not FBOX11, sorry, our LOMP1 flag wild type is able to be highly mitochondrial localized, here visualized in yellow. Whereas this deletion of the MTS region that helps mitochondrial protein get into the mitochondria, we see it really mislocalized here, showing up in red, sort of mislocalized towards the nucleus. Whereas this other deletion here inside the ATPase domain looked a lot like the FBOX11 knockout cells I showed you a minute ago, where LOMP1 still looked pretty close to the mitochondria, but it didn't look like it was getting inside very well. And the quantification of that, we see for the first mutant, about a 90% reduction in localization of that mutant to the mitochondria compared to wild type, and about a 55% reduction for the second mutant. So this tells us that both of these domains are likely, or not domains, but regions of the LOMP1 protein are responsible and essential for interaction between these two proteins. So normally HSCs are glycolytic, but activate their mitochondrial metabolism as they begin to differentiate and proliferate. And as I had told you before, in our normal HSPCs that we depleted FBOX11 in, we could see that there was reduction in these electron transport chain, as well as oxidative phosphorylation signatures. So we evaluated our FBOX11 knockout lines by the Seahorse mitochondrial stress test, and found that both at the basal, these cells' basal respiration, as well as their maximal respiration here, were reduced for the FBOX11 knockout cells compared to when they had been reconstituted with FBOX11. As a proxy for this, by flow cytometry, we measured the mitochondrial membrane potential using TMRE staining, and also saw a reduction in MMP for our FBOX11 knockout cells. Inversely, we took our primary CD34 cells and depleted FBOX11, or now also LOMP1, using 2 different shRNAs for each, and saw a reduction in MMP, as we would expect. So I'm just going to skip through those for time. We came down to this, as I mentioned before, LOMP1 is a mitochondrial chaperone protein. So we took our LCI AML3 cells that we had shown have reduced mitochondrial LOMP1 when FBOX11 is depleted. And we depleted both of these, either FBOX11 or LOMP1. And then it validated the assembly of the electron transport chain that would produce this respiration in MMP. And we could see that in both cases, we had a reduction of electron transport chain complex 4, which would account for these defects. So what we still didn't know at this point is what is actually allowing LOMP1 to get to the mitochondria and properly assemble the electron transport chain here. So ubiquitin is a 76 amino acid protein that is modified through and attached to one another to form these long chains, or can also be attached as single chains to target proteins. So these can lead to all kinds of differential effects, such as targeting through the proteasome, or changes in lysosome targeting, activation, and a whole bunch of chains that still aren't exactly understood within the ubiquitin proteasome system. So we were curious which of these post-translational modifications would be responsible to get LOMP1 into the mitochondria, where it was maintaining this mitochondrial membrane potential activity. So we took our 2 FBOX11 knockout lines, and we, under now denaturing conditions, immunoprecipitated LOMP1, and we could see that under these conditions, LOMP1 bared a K63 ubiquitination mark. So to validate whether this is responsible for the mitochondrial membrane potential, we treated cells with either netylation inhibitor or ubiquitin inhibitor. So netylation is just a different type of modification FBOX11 could potentially add. And we could see that only these ubiquitin inhibitors here on the right were able to recapture this loss of membrane potential. And this is just a validation by Western blot that this is like the drugs are acting on target. So what that now tells us, I know I've gotten really into the weeds here of the mechanism, is that FBOX11 is facilitating this modification to LOMP1. That modification is a K63 linked ubiquitin chain, and that chain is then promoting LOMP1 into the mitochondria, where it can act as a mitochondrial chaperone protein for the electron transport chain complex 4. So we then went back and we looked at these primary samples that I had shown you we'd validated FBOX11 protein expression in before. And we put them in culture compared to, as well as our 25 subject cord blood derived pool of a normal HSPCs. And we could see that there was a reduction in MMP for these cells compared to the AML cells compared to the healthy HSPCs. And we found that there was a correlation for the degree to which the FBOX11 protein was reduced in those samples with the MMP. So the less FBOX protein you have, the less mitochondrial membrane potential. And this also anti-correlated with mitochondrial, with leukemic burden within the sample, as we would expect. So we then did some more work, because why not? Where we looked at some RNA sequencing of FBOX11 and LOMP1 regulation in primary CD34 HSPCs. And we looked at the differentially expressed genes that were shared and different between LOMP1 knockdown and FBOX11 knockdown. And what I want you to appreciate here is that the differentially expressed genes for LOMP1 in this marked by this outer circle here in blue is much fewer compared to the outer circle in red for FBOX11 knockdown, about 1/3 of the total number of genes. So that tells us that FBOX11 is influencing a much wider set of target proteins than LOMP1 is. And LOMP1 activity is likely dependent on FBOX11. And about half of the genes that were differentially expressed in our LOMP1 knockdown were actually shared with FBOX11. And this was also true of the downregulated genes as well. So looking at what those differentially expressed genes, what kind of transcriptional programs they actually belong to, we found that they were shared among those same HSC and LSC gene sets that I talked about earlier. So depleting either FBOX11 or LOMP1 enriched for those transcriptional programs. And it also enriched for defects in the mitochondrial respiratory chain. Inversely, we had the opposite reaction where these knockdowns downregulated those progenitor gene sets as well. Whereas FBOX11 overexpression alone trended these gene sets in the opposite direction to either of the knockdowns. When we take those differentially expressed genes again, and we overlay them with genes that are associated with normal blood cells, we could see that FBOX11 knockdown here in orange and LOMP1 knockdown in blue, they largely phenocopy one another, whereas they are sort of, sorry, I'm not explaining this plot very well. The further out each of the, this sort of colored section extends towards each of these different cell types, the more alike those differentially expressed genes are to those cell types. So we could see enrichment towards HSC, as well as myeloid lineage cells, as well as mature myelocytes. which we had seen in liquid culture before. FBOX11, if we're looking at now the down-regulated genes, FBOX11 overexpression sort of had the opposite effect. And lastly, we had done a combination where we had depleted FBOX11, but also overexpressed LOMP1 to see if we could rescue the effect. And the FBOX11, again, still in orange, whereas the FBOX11 plus LOMP1 overexpression in green, we could see that those two transcriptional states did largely phenocopy one another. And what that tells us is that LOMP1 expression alone is not enough to rescue this phenotype without FBOX11, suggesting that there's a dependency for LOMP1 activity on FBOX11. So lastly, we just validated these transcriptional programs in HSPCs and liquid culture again, just as we'd seen before with our FBOX11 depleted HSPCs. Culturing LOMP1 knockdown cells also had an increase in the CD34 positive percentage after a week in culture, as well as an increase in these G0 cells in the cell cycle. And just as we'd seen before in liquid culture, further along, based on measuring their output, we saw again the reduction in megakaryocyte and erythroid cells at the expense of myeloid bias for both LOMP1 and FBOX11 knockdown. So this tells us that FBOX11 regulates normal HSPCs for their healthy differentiation state through LOMP1 activity. So lastly, the last thing that we did is we looked at and tried to validate these transcriptional programs within primary AML samples just to see if we could recapitulate all of these effects we've been seeing. So there was a paper a couple of years ago that had developed a method where they deconvoluted the hierarchy of an AML sample where you had taken a measurement of the bulk RNA-seq from that patient. And they took 14 different single-cell RNA-seq libraries and developed a signature where they could predict what the hierarchy of that sample looks like. What they ended up sort of describing is they had sort of three major phenotypes where they had this primitive, these primitive cell AML samples where the sample was made up primarily of stem cells, a GMP-like sample where there had been a sort of an expansion of a progenitor-like population, or these mature samples that had more of a classical hematopoietic differentiation hierarchy, which was very steep with very few stem cells. So we took their data, and we replotted this and then separated the samples into high, low, and intermediate FBOX11 expressing samples. And what we found was, when we looked at this, that there was a high concentration of the high FBOX11 expressing samples sort of in this lower left quadrant. And that corresponded to the mature or rather steep differentiation hierarchy. And when we quantified this, we could see, yes, indeed, these immature AMLs with these bulging high population of stem cells and immature cells was enriched for the low FBOX11 at the expense of associating with the mature cluster. So I did get very heavy with the data, so I'm very sorry for that. But basically what this ends up looking like is overall in a normal cell, FBOX11 is facilitating a K63 ubiquitination event of LOMP1 in the cytosol to promote LOMP1 into the mitochondria, where it's able to maintain cellular respiration through its chaperone activity, which leads to a balanced differentiation of a normal healthy cell. Whereas in AML, we see this loss of this FBOX11 protein, which leads to impaired mitochondrial localization of LOMP1. and we get this reduced oxygen consumption and membrane potential, leading to quiescence and aberrant differentiation towards myeloid lineage of these stem cells. So with that, we can thank all of the funders, the patients who allowed us to use their material for this work, everyone currently in the lab, as well as all the previous lab members that extensively contributed to this project. And thank the committee and the proteomics core at BC Cancer as well. And thank you for all your attention today.
That was terrific. Thank you very much, both of you. Are there some questions? Please.
Hi, great talk. I was wondering, so if I understand correctly, it's specific to cells, to specific cells, like this mechanism doesn't happen in other cells, is that right?
We're not entirely sure. I know in literature, a lot of the ubiquitin system, the modifications, the post-translational modifications are very cell type specific. So we're actually not sure whether or not this would be occurring in other cells per se. FBOX11 definitely still can co-immunoprecipitate with LOMP1 within like a HEC293 cell, but we haven't looked in great detail of whether or not this exact mechanism is occurring in other cell types. No.
And have you looked to see where does the protein accumulate outside of the mitochondria?
No, we have not looked at the exact cycle of what's happening to this LOMP1 protein when it's not being properly imported in the mitochondria. We don't know where exactly the protein goes, whether a different E3 ligase is targeting it to the proteasome or it's some other degradation mechanism. We don't know. Thank you.
Well, I have a little question. Can the FBA or FBOX skip1 col1 E2 complex be assembled in a purified system so that it's functional?
I do believe that has been done by other groups. We haven't tried to do any sort of experiments like that. I think because we really tried to keep it within a cell. But I'm sure it is possible. Definitely, it's been done before, but we haven't tried to do it.
Good answer.
Yeah.
I'm curious about this because it seems the way that at least you've drawn your model that FBOX11 is functioning to hold together skip1 and col1. So much like a drug that Ali's aware of called emicizumab, which holds together two proteins in the coagulation system. This emicizumab is functioning as a scaffolding cofactor. We might be able to design a bispecific antibody to hold together skip1 and col1 to bypass FBOX11. Causing ubiquination to go forward.
Yeah, I mentioned it, but maybe I didn't make it very clear because there are sort of 40 different FBOX proteins within a cell that you could have assemble into this complex. So I'm not sure if necessarily just keeping col1 and skip1 together would solve the issue because without FBOX11, you're not going to be able to get docking of LOMP1 to this complex to be ubiquitinated. So if we could find a way potentially to increase the affinity for which the lower FBOX11 protein in these AML cells increase FBOX11's affinity with its complex when its expression is low, that might be able to promote additional interaction between LOMP1 and the E3 ligase complex.
We would still have proof of concept regarding its cofactor function within the direct ubiquitinase complex. Anyhow, Interesting. Protein chemistry, as Ali says, may be fun to talk about more over a beer, maybe 6 beers.
Yeah.
Thanks very much, Hale.
Yeah, thank you so much.
More questions? Thank you very much for visiting.
Yeah, thank you so much, everyone.